Nice, a long line of yeses. I think we're good to go.
Okay, perfect. So, as I was saying earlier, this is a presentation about AlphaFold, especially AlphaFold 3. We will not go like in technical details better, too much in technical details. This is like perfect, I would say, for starters and maybe new buys in fields. We will have, as I listed here in the Outlook, a look at what proteins are and how do we get them. Okay. We will understand what is the protein folding challenge and why it is so important nowadays, and why it has been in the past. Then we will move on to understanding AlphaFold generation, let's say, so AlphaFold 2 versus AlphaFold 3. We will see, this is a little spoiler, the diffusion architecture in AlphaFold 3, new tasks that AlphaFold 3 is able to perform with better accuracy. We will go through the known limitations of the software, and then we will have a little bit of a conclusion, and then we will have like open questions. We will see open questions that AlphaFold let us read. Then after that, we will have a rapid Q&A session. So as I was saying earlier, just keep your questions for the end, and I will like try to answer if I can. I will try to answer. So let's start with like the very, very basics. Okay, so what are proteins and how do we get them? Okay, so you have to imagine proteins like the all doers in our cells. So they do everything, they build up our existence. Okay, they are our immune system, they are they are in our muscles, in our brain, they carry messages in our body, and they do all sort of stuff. They are our hormones, everything you can think of. Maybe a protein does it. Okay, and especially obviously if it is a biological function, for sure. So proteins, the journey of proteins starts with DNA. Okay, so you probably all heard of DNA. DNA is like the source code of life. Okay, if you ever imagine a programming language in which life is written, that language is probably DNA. And as you can see here, there are 4 basic letters of DNA, which are A, T, C, and G. Okay, so these letters are used to encode and then are translated into these other letters here. I don't know if you can see it. I can't. But these letters are
so that we can see the letters. Can you put it like full screen or presentation mode?
Yeah, I can.
Perfect. Yeah, that's a lot better. Nice.
So, as I was saying, you can see. Okay, now we can see all the letters here. So as you may notice, these letters are a little bit different from the ones in the DNA, because the ones in the DNA are nucleotides. Okay, the building blocks of DNA, and the ones in proteins are called amino acids. Okay, these amino acids are actually the building blocks, the bricks that make up a protein. Okay, and there are 20 of them, which are encoded by sequences of A, T, C, and Gs, and then translated by little operators in our cells that are called ribosomes. Okay, this is like basic biology, and probably every one of you has heard it in like high school or stuff. From the translation, you get what is called the primary structure, so an amino acids chain. This amino acid chain is a linear structure, so you can imagine it like 1D structure, one-dimensional structure. After that, you get on, the protein gets a little bit folded, a little bit worked around, and you get the secondary structure. As you can see here, we have like these beta sheets and alpha helices that are not like that important. Just so you know, these are like the most common secondary structures. There are plenty of others. We will not discuss them. But this is like the basic way in which proteins get from like the primary structure to the, let's say, mostly 3D, but also partially 2D structure in the secondary portion. And that like the portion of interest to us comes when the protein gets translated from the secondary, it gets like built up from the secondary structure to the tertiary structure. What happens here, nobody knows. Okay, so even the cell doesn't know what to do, actually. It is a process of trial and error, which is repeated thousands of times, okay? And you have to imagine that all of these happens in a fraction of a second, not even in a second, in a fraction of a second. We're talking about milliseconds, okay? All of these, and this happens millions of times in your body, each second passing. Okay, so this is fantastic on one hand, but on the other hand, a terrible hell for bioinformaticians, because we had a great challenge. Is there any problem? Can you see everything?
Discord?
So, as I was saying, the big problem is like, how do we go, like let's say, in a computer way, because this is a challenge that's haunted by informaticians. How do we go in a computer way, computer language, from the amino acids chain to the, let's say, three-dimensional 3D representation of the protein? Okay, so this has been like a hell for, as I said, a hell for bioinformaticians. They tried everything they could from the 70s on, from when the first laboratory, like chemical, biological stuff was available to see actually the protein, to see actually the real protein structure. They tried to simulate it with computers, and they didn't seem, and they can't seem to get it right. Okay, so there wasn't a reliable algorithm that let bioinformaticians and people interested, protein scientists and whoever was interested, to turn linear proteins, okay, linear sequences of these little letters here to three-dimensional structures. Okay, and this was a big problem because we need to know how a protein is structured, for example, to design our drugs, to design our, let's say, vaccines, or to design targeted treatments for cell diseases, okay, cellular diseases. So whatever interacts with your body, whatever drug you've taken in your whole life probably has interacted with proteins in your cells. If you have ever taken an antibiotics, for example, antibiotics mostly interact with proteins, okay, in bacteria. So if you don't know how the protein you are going to target is structured, you can't design an effective drug, okay, an effective treatment for that protein. Okay, so you can actually see it in your laboratory. You can actually try it in your laboratory, but it's really time consuming and it costs lots of money. Okay, at least it costed lots of money back in the days. Okay, so they were trying to find something which was cheaper and faster, and they were trying to do it with computers. So AlphaFold came in. Okay, and AlphaFold came in really recently in 2020. The first AlphaFold made the first appearance. So just a little bit of background for any one of you who may have never heard of AlphaFold. AlphaFold is a, let's say, partially machine learning, now partially AI software, which was developed by Google AI spin-off, Google DeepMind, for the protein folding challenge. They had their first test version tested out in 2018, and the actual version, the one that gave the best results, was AlphaFold 2 in 2020. And as you can see here, with our little cocked cat, it was a real shock for everyone when in KSP, in CSP, in CASP 14, so the Critical Assessment of Structure Prediction 14, the 14th edition, in 2020, AlphaFold came in and scored a neat 92.4 overall accuracy. Okay, or global distance test, but let's call it accuracy. Okay, for let's say. And you know, as you can read here, a score of 90 is considered competitive with laboratory procedures, which means you have like you can get the same results from your software and the laboratory, okay? And laboratory is considered golden standard, okay? So whatever you get in laboratory means it probably is right, and whatever you get with software probably is wrong. So you have to compare the two, okay? So AlphaFold scored a neat 92.4, which was like shocking because no one had surpassed a score of 60 in no previous edition of CAS. Even AlphaFold, okay, so the base version of AlphaFold 1 or 0, if you want to call it like that, did not surpass 60. And now, in just 2 years, Google DeepMind came in and like crashed the stage with AlphaFold 2. And so, the excitement of bioinformaticians at that point was like peaking, because they thought, okay, we're done. We solved the challenge that like haunted us for 50 years. We can now predict all existing protein, and that's actually what they did. They predicted 200 million protein structures, which are like, I would say, pretty much all the known proteins in the world. So, and you can search them. You can go on AlphaFold database. You can search for these proteins, and maybe, if you're lucky enough, you may be the first person to see that protein structure, because most of these proteins, no one have never seen them. So, just give it a shot, and maybe you will turn out to be, the first person to see the structure of the protein of some kind of exotic bug that is in the database, and that AlphaFold, for some reason, predicted the structure for. So just give it a try. It's really fun. It's really easy, and it's really also user friendly. So you just need to type in your browser AlphaFold database, and you will get big, huge database. Okay, so 200 million proteins, and up to that point, okay, so up to 2020, we were just able to predict like something like 20,000 protein structures. So you can see it's like a manifolds advancement. It's like orders of growth, orders of magnitude bigger than what we were able to do before with laboratory techniques in just 2 years. So that was even more shocking, okay, on Google DeepMind sites. And so we now have to like little bit have a look to the structure of AlphaFold to understand what is going on. So I won't go, as I said, in too much detail, but you can see here, you start with an input sequence, you then and then the process branches out to two main approaches. One that is based on the genetics, and one that is based on the structure. Okay, so on the genetic side, you get what is called MSA. MSA stands for multiple sequence alignment, and it's, as you can maybe see here, it's like taking all the protein structures we know from all the species. For example, here you can see the human species, chickens, lizards, and things like that, and we compare them in a multiple sequence alignment. It's like a matrix of little letters compared and aligned between them, among them, okay? And on the other hand, you get another matrix. So here in like the blue one, so as you can see, the pair representation one is a matrix that correlates each amino acid with the following amino acid in your protein chain, okay? Each amino acid with the following amino acid, which can give you like some positional information, some positional metadata, which go into this like evoformer block, okay? This big block, this evoformer block. I won't explain how it works specifically because it's too technical, but just saying it compares and reiterate this predicted structure based on several reiteration of MSA to pair representation comparison. Okay, so after that, you have like your two matrices, your still the corrected MSA matrices and pair representation matrix, and you feed it to the last portion, which is a structure module. The structure module takes in all the metadata, all the positional things that you can imagine, all the chemistry, all the pre-instructed things that the training dataset also had, and obviously the data also have, sorry, not the training data, but the data also have it with them, and predicts a first three-dimensional structure. And as you can see, there's a recycling for these let's say cycle for 3 times. At the end, you get a three-dimensional structure. But this is, as you can see, not based on any specific, for example, diffusion, or any specific diffusion, so image generation, for example, or 3D image generation, but it is pure, let's say, learning and machine learning stuff. Okay, integrated with some AI, but pure machine learning stuff. And it's computationally expensive on one hand, and okay, not as low as laboratory techniques, but still slower than what we can see next. Because now we are going to unveil what is the main character of our presentation, which is AlphaFold tree. Okay, so, and as you can see from our cute cat here, AlphaFold tree has a really more, let's say, easy and understandable and also effective architecture. So you have like pretty much the same thing at the beginning. You have, let's say, in AlphaFold 3 authors, AlphaFold 3 paper authors just say that there was like some slight changes in the first blocks. And what is really different here is that you can see here at the end, you have the diffusion module that actually is a generative AI module that leverages diffusion to predict the 3-dimensional structure of the protein. So here GenAI comes into play. And let's see what is actually this stable diffusion, or this diffusion architecture. So diffusion architecture here is not like the basic, like, let's say, text to image diffusion architecture. So you don't go from we're not at the point that we get an amino acid sequence and we receive out we input an amino acid sequence and we receive out from the stable diffusion model a 3D image. But we have like a guidance input, which is, like, as you can read here, a fuzzy image, okay, that is like received from the upstream network, okay, from the blocks that we see earlier that actually make up AlphaFold. And this fuzzy structure comes with like amino acid coordinates, chemical relationships, physical relationships, all this stuff. And the stable diffusion model is now able to like generate variations of this fuzzy image, 3-dimensional real structure, real variations of this fuzzy image that represents like a distribution of the possible, all the possible, or let's say lots of the possible 3D structures of the protein we have. Okay, so even though let's say the upstream network is not that confident in the position of specific amino acids or the overall organization of your protein, the stable diffusion model is able, by producing like this distribution of solution and by choosing the most probable one as your result, is still able to capture most of the variability and to give you a reliable answer. Okay, obviously this is a great advantage, but you know, as every Gen AI model, it is like prone, and I won't say prone, it is like possible that it hallucinates. Okay, it has hallucinations. So this was something that Google DeepMind scientists encountered, and they said specifically in what are called unstructured protein regions. So these are regions that even in the biology actual structure, in the biological, in the cell, have not a three-dimensional structure, have not a defined three-dimensional structure. So you can't get out something that is defined. So the model obviously is instructed to try its best to have, let's say, to get out a structure. So it turns out into like this incredibly random, but also like, you know, reasonable structures that actually are not, okay, that actually are not real. So what did they do? They took like other unstructured regions, other unstructured, let's say, proteins, okay, that they generated with another AlphaFold version, which was specifically trained on that task, so on recognizing and producing specific patterns for unstructured regions. And now, when it comes to unstructured regions, AlphaFold 3 is able to recognize them, and when it comes to the stable diffusion portion, it just produces some random loops. Okay, so this loop structure, some coils. Yeah, you know, the technical term for it, it's random coil, and means you can like hear from this random coil. It seems pretty much unstructured. Okay, so it's definitely unstructured, and you can easily, you know, recognize it and see, okay, AlphaFold 3 predicted an unstructured region here. Okay, obviously, maybe not at first take if you are not a protein scientist, but you know, with some expertise, you may be able to say, oh, okay, yeah, this protein is renowned for having like unstructured regions in itself. So, okay, probably it is an unstructured region. So this, so far so good for the diffusion architecture. I will switch to what actually is AlphaFold doing new. Okay, because so far we talked about protein folding. Okay, so AlphaFold is able to fold proteins. Okay, not big news. Also, AlphaFold 2 was able to do it, and it was able to do it like really well. AlphaFold 3 seems to do it like better because of this, especially because of this stable diffusion architecture at the end. But this is not all that AlphaFold 3 has to like offer us. On another hand, you get three special fields that Google DeepMind underlined as like breakthrough field for AlphaFold 3. The first is protein nucleic acid interaction. So to put it simple, nucleic acids are our DNA and RNA. Okay, DNA is the source code, and RNA is like the compiled code. Okay, is a code that ribosomes, which are the small little factories that produce proteins, can understand to translate it into amino acids. Okay, so these are pretty much really similar proteins and molecules, and proteins can actually interact with them. Okay, so you have like in this moment, your brain, your hands, your stomach, whatever you are, is going through like tons of operations. Okay, and in all these operations, the information that comes from DNA is involved, and what helps DNA enacting the information are proteins. Okay, proteins are what carries the information. Proteins are what helps translating the information. Proteins are what actually do what they are told by DNA. So protein continuously interacts with DNA, and also RNA. And so as you can see here, you have like lots of tasks that you can address when predicting protein nucleic acid interactions. You can, for example, use it for genome editing. I don't know if you ever heard of like CRISPR-Cas9. It is like the cutting edge genome editing method, and it uses a protein to cut DNA and to insert new DNA, for example, to cure like diseases. I don't know if you ever heard of sickle cell disease, or if you know someone that has sickle cell disease. If you know someone that has sickle cell disease, chances are that not too far in the future they will be cured with CRISPR-Cas9. And let's say AlphaFold 3 may be helpful in building a specific protein, specific Cas9 protein, because the protein is known as Cas9, specific Cas9 protein that is able to help your friend, your acquaintance, to get cured from, let's say, their condition, their sickle cell disease, or every other condition that involves monogenic diseases, like as, I don't know if you ever heard of Duchenne muscular dystrophy, or other, let's say, other diseases, maybe also some cancer, some tumoral diseases that they will be cured with genome editing, and they will be cured, for example, also by other, let's say, interaction predictions that we will see just in a minute. Okay. On the other hand, there is also this big challenge that we are facing right now, and Google DeepMind is like closely working on it on other sides of its research teams, which is antimicrobial resistance. So you know about bacteria, and they are being so resistant to antibiotics. So there are some statistics that say that every antibiotic that is rolled out now in 10 years will be completely useless, because bacteria will be able to, like, go around and infect us anyway, independently of we taking the like antibiotics. Okay, so this will be a huge sanitary challenge. And if we have some reliable methods to predict the interaction between the proteins and the DNA of microbes, the protein and the DNAs of bacteria, we will be able to real time develop drugs that will be reliable, that will be strong, and that will be effective. Okay, no matter the antimicrobial resistance, we will always find a way to defeat microbes if we have some reliable protein nucleic acid interaction prediction models. Okay. On the other hand, we can have like this second task, which is protein ligand interaction. I don't know if you have ever heard this second word, ligand. It comes from something that is like really similar to my mother tongue. Ligand, it's coming from the Latin ligo, which means to bind. Okay, so this means this is like the binder that it's like accepted by a receptor. Okay, you have to imagine, for example, a little molecule, a little small tiny molecule that gets inside a receptor. Okay, this is the ligand, and the receptor is our protein of interest. And this is like fundamental when we want to do drug design. For example, we have developed a whole new class of, in the 60s and the 70s, people have developed a whole new class of like drugs, which are called the selective serotonin reuptake inhibitors, that were actually a breakthrough in the cure for physiological and major depression. And this was actually the path they used. So they searched for the receptor. They saw what was going on with serotonin and its receptor, and they said, okay, we now need to find a way to help people with these like receptor defection or uptake defections to inhibit these mechanisms. And how they did it, they designed molecules to do it. And up to now, we use these docking methods to design molecules. Docking is like taking a molecule, placing it into your spot of interest in the protein, and try to simulate every possible position, every possible interaction, with also every possible, let's say, variation of your molecule, okay, of the drug you are developing. And docking is really computationally intense. So if we have something that is reliable and fast, and also computationally light, like as AlphaFold 3, we will be able to deliver drugs really fast, and maybe also with higher accuracy. Because as it happens, AlphaFold 3 outperforms every docking, let's say, tool that we have up to now, okay, that was ever developed up to now. And on the last hand, we have antigen-antibody interaction. Antigen-antibody is key when it comes to your immune system, okay? So when you have like a pathogen, a virus, or a bacterium entering your body, your immune system generally recognizes it and blocks it. And it does so by producing antibodies. These are small protein molecules, okay, that binds to specific region of your virus, for example. Don't know if you heard of, probably you did, of that spike protein for COVID, for like coronavirus. This was actually what we here call as antigen. Okay, antigen is like what comes from the external world in your body, and the antibody is actually what fights, what is able to recognize and fights back the antigen. Okay, when it has recognized the antigen, it calls for the big guns. Okay, it calls for the cops or whatever, the soldiers or whatever you want to call them. They're your immune system, and they can recognize the pathogen and kill it, okay, and erase it from your body. This goes on every day in your body, even though we don't even like notice it. And this isn't just key in recognizing pathogens, and also, for example, designing vaccines that are not immunogenic, that are not interfering with our immune systems, but also it is key in recognizing tumoral cells, because tumoral cells are little, let's say, are sneaky. Tumoral cells are difficult to recognize because they are slightly modified so that your immune system doesn't notice that you have that tumoral cell in yourself. Okay, and if we have some sort of tool like AlphaFold tree, which is able to automatically recognize your tumoral cell, your mutated protein's tumoral cell, we will be able to identify it quicker, and we will be able to create some better, let's say, drugs and stuff to let's say cure it. Okay, so this is so much for now. I will just start talking about like the limitations we have. No limitations are the model struggles to predict, let's say, chirality. Chirality is a core concept in protein science. Chirality is like how molecules are rotated. Okay, there is also a problem with clashing atoms. Clashing atoms are like, it means that the model predicts atoms that are superposing with other atoms from the protein, and this can happen because, okay, if an atom occupies a space, another atom can't be there. Okay, and so this is a little bit of a struggle, especially when it comes to long strings of DNA interacting with large proteins. And also we have the problem that we already mentioned of hallucinations, but also obviously DeepMind tried to mitigate these issues. Okay, also there is some room for improvement in the paper. In the discussion of the paper, they say that this antigen antibody, let's say, architecture is problematic and is some way problematic, and they try to get like big results they had like better results with more seeds. So seeds are like, let's say, I would say random strings that actually help you predict the let's say help the model predict the image or generate the image. So these are strings of number, or let's say it's chains of numbers that are given to the model and that instruct the model in predicting the protein or the protein three-dimensional structure. But also we have like some other inherent, let's say, some other internal constraints which are like internal to every model, to every, like, let's say, to every protein folding model, because every protein folding model that we invented up to now is able to predict only static proteins. Okay, so proteins that are not in a living cell. And this is a big issue because our living proteins are in living cells, are in water solutions, are in our cytoplasms, and so on. And so obviously we need to make some little adjustments when it comes to these kind of proteins. Okay, so we had to and Google DeepMind say they are trying also to get this problem sorted. But it is difficult because all the data we have available come from proteins that are no longer in their biological environment, okay, that are fixed, that are static, and this can be a little bit of a problem. So just to, let's say, conclude all this, let's say, all this talk we had, we have like, AlphaFold 3 is like a breakthrough, but it's not a finish line. Okay, this is not an endpoint. This is the starting point, the beginning of a new approach to complex biological problems. Okay, complex biological problems are now being addressed with generative AI. And AlphaFold 3 is not the only one doing it. Lots of other tools that are being developed. For example, don't know if you heard it, but there is also this other really interesting Gen AI application, which is Open CRISPR, which is used for genome editing. Okay, so if you're interested, you can look it up. It's really real fun, and what is most important, it's open source. Okay, there are also some, let's say, to conclude, let's say there are also some explainability and transparency concerns linked to AlphaFold. Okay, because the increased use of generative AI improves the accuracy of our results, but let's say diminishes the explainability. Okay, so you don't get to know why a protein is like this. So the diffusion model produced it, and you can't see. It's like a black box problem. Okay, it's pretty much it. It's pretty much it. And when it comes to designing proteins that may be key in curing diseases, this cannot be. This actually cannot be, because you have to know why a protein is like this, and you have to have some kind of support. And AlphaFold can give you all the metrics that it wants, but you always need, let's say, some sort of more in-depth explanation. And with the introduction of generative AI, this is going not, let's say, this is going not really well. And also, there is also another big issue with transparency, because Google DeepMind released a sort of like mocking version of the code. This is not the full code, and they won't release the full code. They said it expressly. They won't release the full code. For now, AlphaFold 3 is also limited access for researchers and normal users can't use it. In with unlimited access, you have to be a researcher, you have to be connected with some sort of institutions, and even with that, AlphaFold comes with limitations. I'm not excluding that Google DeepMind and generally Google will release versions of AlphaFold that will be available to everyone, and we will be able to use it unlimitedly. It will probably be a matter of time, but the actual code, with which normal developers can try, test, and also maybe improve, contribute to the development of AlphaFold, is not available and will not be available. So this raises some questions, some open questions that I'm probably asking you to think of when this presentation will be off, when you have in your day, today, tomorrow, in the future. So as generative AI, or generally AI, is becoming an integrated part in our lives, we are facing the question: are we really interested in having an algorithm which is possessed by a big company, a big tech company, and which is not open source, deciding for our drugs, for our treatments, for our therapies? Are we really willing to do that? Or do we want something like that is accessible to everyone, that everyone can improve, that everyone can test out? Obviously, there's no right or wrong reason here, there's no right or wrong answer here. But I will just try, and I will just invite you to tease your brain on these kind of questions, because they are becoming every day, and I'm sure every one of you has already heard these kind of questions. They are becoming every day more present. They're becoming every day heavier on our, souls and conscience as developers, as AI developers, and we owe the users, and we as developers, or we as developers owe the users the rights of being explained what is going on with the algorithm, with everything with it. Okay, so, and we as users would want the same thing if we were on the user side, and if we will be on the user side, when we will be, not because it's not a matter of if, it's just a matter of when we will be on the user side, we will maybe, I personally will maybe just a little bit scared and questioning what is happening, why is happening, because obviously because I'm like most of a why person, but also because we need explanations, we need to know, and open source would help. Sadly, AlphaFold isn't open source. So just a little tip, I have here like uh, um, let's say a little bit of a demo of a space, of an againphase space I built that leverages an open source protein prediction model, which is ESM2 by Facebook Meta. And this protein prediction model, as you can see here, is able to predict the protein structure from, you just input your amino acid sequence, so your letters, and you get out the structure. You also get, so you can see here the static image, but you also get to download the 3D model, and you can play with it. Okay, as you can see here, you can just go around and play with the model. This is just a start. We will implement more like in-depth explanation, more metrics, or even the metrics, and also all sort of stuff. And we will be probably able to also build some in-browser 3D representations of the protein. You can play on the browser. For now, it's just like a start, but it is already something which is open source. First of all, open source, which is fast, and also pretty much reliable because it leverages a big model that outperforms AlphaFold 2, that is kind of midway between AlphaFold 2 and AlphaFold 3, okay, ESM2, which stands for like evolution sensitive model, and it's an evolution based model for the protein. We didn't get like these, but it's just, it works through the evolution of protein. Because obviously also protein evolve, life evolves, and so proteins evolve too, and this model is able to predict the three-dimensional structure of proteins based on their evolution. So I'm done. Okay, so this is the end. If you have any questions, please feel free to ask. I will now, let's say, dismiss my screen presentation, so I will be able to see if there is any questions, if there is any raised hand, and obviously I will try to answer your questions. I will try, obviously, and just, thanks for listening. Many thanks for listening, and you will find all the references for the articles and for the papers here at the end of the presentation, but they are pretty much the ones that you already got in the reading group channel. So I'm dismissing my screen presentation right now, and yeah. So how do I do it? Okay. So if anyone has other questions, or every sort of question, there is no wrong question, there is no stupid question. Okay, no such thing of that. I will be really pleased to answer.
Thank you a lot for the presentation. I had one question about the diffusion part. So like I think like amino acids, as far as I know, there's like a wide range in like the size they have. Like they can be like 10 times the size of like some other amino acids. But usually when I hear about diffusion models, I know that they struggle a lot with like resolutions outside the size they trained in. So like, is there like something there where like something creative they did to like account for like how to like adapt the like big amino acid sizes like that is out of distribution during training?
So I don't know if I fully understood your question, but you are asking here if they took into account the varying dimensions, the varying like 3D dimensions of amino acids, right? In their training, did I get it right?
Or like kind of like the length. I feel like the biggest like protein can be like I don't know, like around 23K amino acids or something. So I was thinking like I was pretty impressed that AlphaFold 3 is able to like account for sizes that large, because usually diffusion models can't do that, I thought. Yeah.
Yeah. Okay. Yeah. Actually, there is a reason for that, and generally the reason should be found in the fact that not everything is done by the stable diffusion model, but there is, a portion of it that is done by neural networks. Okay, basically neural networks that give the stable diffusion model, sort of a draft image. Okay, so you get a draft image, a guidance image, and all the stable diffusion model gets to do is using this draft image to produce distribution of solution, out of which the most probable is selected. Okay, so the accounting for the length and the dimensions of the protein is pretty impressive, and already AlphaFold 2 was able to do like really big proteins. Okay, and this kind of thing is now pretty much improved by the fact that you have this guidance image. Think of it as like a black and white image that the stable diffusion model is able to turn into colors. Okay, that's pretty much it.
Okay,
Don't know if I answered your question.
I don't think I fully get it, but kinda I think I got the intuition. Thanks.
Okay.
So you mentioned that AlphaFold 3 works better with ligands. Does the stable diffusion model like just insert ligands on its own without me putting it obviously I have to submit a sequence of amino acids to it, and it's going to tell me what it looks like as a structure. Do I also submit what ligands I think are in the protein, or the stable diffusion model will like generate likely ligands that get inserted into the protein?
Okay, as far as I know, because obviously the model is not, you are not able to predict ligand protein interaction yet on the AlphaFold server. As far as I know, you have to input the protein structure and separately the ligand structure. Okay, or you have to specify the type of ligand if it doesn't have a protein structure, because obviously most of the ligands are just like chemical, just like think of adrenaline. Adrenaline is a ligand, but it is a small little tiny molecule, which is nothing to do with proteins. So you have to just specify, for example, the structure of the ligand. You get to do it with like chemical 3D models of molecules, and then you feed it to AlphaFold 3, and hopefully you get out the interaction prediction. But you have to feed them separately. So as far as I know, I repeat, this is still not available. It will be in the future, I hope. And ligand obviously has to be submitted separately from the protein sequence. Sorry,
right,
from the protein sequence. Thank you.
There seems to be a question in the chat on like, yeah.
Oh yeah, right. Obviously, I wasn't checking the chat. So can you elaborate on how accurate the protein DNA complex prediction is? For AlphaFold, you mentioned the GDP, which is GDT, or GD, okay, score that was comparable to experimental accuracy in the prediction of protein DNA prediction, now also as accurate as lab experiments. Yes, so it is really good. Now if you go through AlphaFold paper, you will see that they all outperform already existing tool, which had already comparable results to laboratory procedures. And also, I would say that there are not really reliable, let's say, really reliable methods to predict protein DNA interaction in laboratory. Okay, so I'm most dry lab stuff. I'm most computers, but from the friends that work in, let's say, wet lab, so that works with all the cells and things like that, I would say that this is like really difficult. They have to do it with fluorescence marking and all that kind of stuff that is time consuming and stuff. And we already had, as I said, we already had tools that were really good at predicting the structure, and AlphaFold 3 is even better. So yeah, it outperforms already existing models. I do not have specific metrics on hand right now, and I do not remember. But if you go through the paper, you will see all the images, all the bar plots, and all the stuff you need to know at the end, and you will have the comparison and the metrics and things like that. I don't know if I answered your question, but you know, and so there is like a question by self-taught swimmer, which says, I don't think Cas9 is used directly to edit cancer cells. I think it will mostly use directly edit cells that are used to manufacture neoantigen and unique to each patient's cancer cell. Yeah, that's neat. Absolutely. The end goal for this could be personalized neoantigen vaccines. AlphaFold 3 could speed up the process, I think. Yeah, you know, you're right. You're totally right, self-taught swimmer. That's perfect. So, okay. Don't know if you have any other questions. Just feel free to drop by. I would say I would answer just one more question because I need to rush to dinner because they are closing the canteen. So another question: can AlphaFold 3 also be used for designing new proteins? Actually, this is an odd one. This is a tricky one. Yes and no. You can use AlphaFold 3 to predict new proteins, new unseen protein structures, okay, but they are existing protein structure, because still AlphaFold 3 is based on, you know, existing structure and evolutionary links for these structures. So it is still difficult generating new proteins. But if you get like a new protein which is like natural, which seems a natural protein, you can sort of predict what it will look like in a static environment. Okay, so you can, as far as you design a little small protein which is similar to the actual existing one, but you can't on a very large scale. Okay, if you're interested in designing new proteins, especially Cas protein, I would suggest, and I would write it somewhere in the reading group, I would suggest checking out the OpenCRISPR initiative. OpenCRISPR is able to design new proteins. Up to now, AlphaFold 3 is kind of, but it is not specifically targeting new protein design. Okay? So I'm really sorry. I will need to run. But if you have like any questions, feel free to pop by my DMs or on the reading group, just like mentioning me, and I will be super glad to answer your questions. Okay, I'm really sorry, but
Sounds great. Thanks. Take care.
Thank you.
Thank you very much for the occasion, and just real thanks for everyone for being here. Thanks Adam, thanks Chal. Real thanks. So
Yeah.
see you.
See ya.
It was really nice.
See.
Thanks
Bye.
everyone for stopping by. And if you're working on anything in the future, or even if you have any papers you're interested in, hey, let us know and feel free to present. Take care.
Bye. See ya.